John?
That's fine, no worries.
No problem. We are recording now, and Sean's going to get us started. He's got a lot of information to share, but he's only going to share part of it right now. We're considering having a two-parter for him. And if anybody has any suggestions on how to do these better, I'm up for hearing all ideas. So take it away, Sean.
Yeah, I can't share. I'm not enabled to share.
That might help. Okay, can you now?
Yeah.
There we go.
Cool. Okay, everybody. I'm in the chemistry and department in the Machinina lab, but my co-mentor for the interfaces program is Dr. Elizabeth Villa. But for like an aim way down the line from the stuff I've worked on so far. And yeah, I just wanted to show you guys what I do. I study phosphorylation and acetylation of a protein called mitochondrial transcription factor A. Yeah, and a lot of my work, unlike all of you who are either doing, stuff on like, between atoms or basically or like the brain or the heart, I kind of inside the cell, and actually I do most of everything in a tube with purified components. And I study this structure called the nucleoid, which is actually mitochondrial DNA plus all of its proteins. So yeah, you have mitochondria in your cell, and inside the mitochondrial matrix are these nucleoids. And I study like two main components, or two main processes, I should say, in the nucleoid, which are shown here on the right, either packaging of the nucleoid by this protein, Tfam, the mitochondrial transcription factor A, and I study the transcription initiation because it's a piece of DNA you have to transcribe. So yeah, my agenda ideally today would be to get through the background and the I've done so far. But I timed this yesterday and it's just not possible basically to get through the background in 20 minutes, or I'd have to have no questions to get through it, maybe in 10 minutes, but highly doubt that will happen. So yeah, I'm hoping to get through the back and then, yeah, maybe guys what I've done later. So that's kind of unfortunate, but yeah. Okay, so this is just another view of like what I'm talking about. Some people don't even know we have mitochondrial DNA, which is pretty weird when we think about it, why we even do. So in a cell, one of your cells, you have nucleus and you have DNA in your nucleus, and most proteins that are or that run your cell are coded for in the nucleus. And actually the mitochondria needs a lot of proteins, and you encode a lot of proteins that get transported to the mitochondria. So 99% of all the proteins that mitochondria uses are actually encoded by your cell in the nucleus and get transported there very specifically and imported and then used. But you have some genes that are actually continue to be in the mitochondria on this mitochondrial DNA, and every single one of them is used in oxphos complexes. So most people, just freshman biology, know that the mitochondria is the powerhouse of the cell. Whenever I talk about what I do, I get that response pretty much immediately every time. So yeah, that's its main function, right? Are these oxphos complexes generate ATP that then can run pretty much every process in the cell. But the mitochondria do a lot more. They also oxidize fatty acids when you need them, and those, the products of that get used in oxphos as well. But they also make heme, which is super important, and everybody's heard of heme. And so yeah, they're pretty busy. There's a lot of things the mitochondria do, but that's just a general primer of how they're pretty integrated now into are cells, and they used to be an ancient bacteria, which is why they actually have this DNA to begin with. It's circular and compacted a lot like bacterial DNA, which is pretty cool. So yeah, anyone can just stop me if you have a question, but I study that DNA. So yeah, what does the DNA look like? Here it is. It has 13 genes, and you see the products of these 13 genes go directly into these oxphos complexes, and they're generally thought to be pretty hydrophobic. And the reason that you have to maintain this DNA, and because this old bacteria had all of its own DNA, and over time, nucleus actually taken over the duty of all housing all those genes that these mitochondria use. But these genes really couldn't get moved out of the mitochondria. And the idea is that the proteins they make are so hydrophobic that they actually have to get translated right at the source of where they're going to be used. So basically, they're translated ideally inside the mitochondria by a mitochondrial ribosome right into these complexes, because transporting these proteins, the hypothesis is that transporting them would be pretty, I guess, either deleterious or like not possible for these proteins. Yeah. Yeah. You have two promoters for mitochondrial transition to get these genes made. And they're both in this one small little section of the DNA. And the DNA is so it's like 16.5 kb. So if any of you have ever used a plasmid, many plasmids we use in the lab are like 2 to 6, maybe 8 kb at most. So it's kind of just a glorified plasmid. So really big plasmid, basically. And some other cool things. I don't need to go too in depth about this, but yeah, it encodes their own tRNAs for use by mitochondrial ribosomes, and the two ribosomal RNAs for the mitochondrial ribosome are also on the mitochondrial DNA. So, any questions? Are we good?
Yeah, Sean.
What's up?
Sorry if this is a dumb question, but I maybe I'm forgetting my basics of biology. Why is being hydrophobic like stopping these proteins from being transported through membranes?
Well, oh my gosh, yeah. That's probably more of a question I probably can't answer with it because I don't know enough about transporting into mitochondria, to be honest with you. But I think that like intermembrane proteins will like actually have hydrophobic residues on their like surface, probably, I'm guessing, to be inside of a membrane. And that's not the normal way a protein folds. And so getting through the cytosol in that shape is probably not very easy for them. So yeah, that's why the hypothesis right now is they like have to get translated as they're probably translated as they're entering the membrane, like itself. And so yeah, that hopefully that answers your question.
Yeah.
Okay, cool, cool, cool. So I study the package of the DNA, and the star of this show is this protein called Tfam. And this like picture here kind of shows how Tfam would work. Tfam kind of binds DNA and can bend DNA. So this is from a crystal structure. And Tfam kind of like honestly causes a 180-degree turn DNA, which is pretty gnarly. And a whole bunch of Tfam binding this DNA forms this ball. So these are electron micrographs of like kind of making the nucleoid small, basically, because you need to save space. That's one of the main reasons. And also, you need to protect this DNA. The hypothesis is you to protect it, which is actually stuff I'm working on now. But yeah, I guess the big question is, and the question I'm trying to tackle is, how does TFAM know when to come off, when to like, be a little more loose? So maybe a loose configuration or less TFAM around to allow processes like transcription and replication of this DNA. Because if there's too much TFAM, how can any other, enzyme, how can any enzyme get onto this DNA to do anything? So there's got to be pretty careful regulation of TFAM. So yeah, hopefully that all made sense. Okay, just another quick look at Tfam, because my project does rely on knowing some of the portions of Tfam, and I'll probably have to show this again some other time when I show my actual data. But just the general gist of this is that Tfam has these two L-shaped domains. Without getting into too much depth, it's actually kind of a simple protein, right? It's got this L right here, and this L kind of backwards L looks really similar to this L. And in between them is just a linker, something to stick them together. Yeah, and these L, there's a leucine residue that actually kind of embeds itself between the bases and causes some local melting of the DNA. And that local melting gives the DNA this flexibility to be bent this tightly. So that's pretty cool. But besides that, Tfam also makes some, either down here is a base specific contact, and here is, between the ribose portion and yeah, threonine, some hydrogen bonding. So it makes a lot of contacts along the way with DNA. And the linker probably makes a lot of contact with the backbone, which we all know is negatively charged, right? That's kind of important for my project. And so, yeah, I guess what I try to tackle, again, I'm just going to reiterate it because it's going to come up. But right now, the current theory is that post-translational modifications, which is when you actually modify a protein that's already been translated with something like a phosphate group or an acetyl group. Other ones are ubiquitin, is another type of PTM you guys all might have heard of, but you can maybe modulate how Tfam binds DNA other than actually having to degrade Tfam, reduce Tfam you're making, right, from the nucleus, because Tfam is actually made in your nucleus. Well, the genes in your nucleus is made in the cytosol, but yeah. And the idea would be that if you add groups to these, L-shaped domains, you would maybe change how Tfam binds. So yeah, even to make matters more confusing, this is exactly why I can't get through all my background. Tfam has another role that's obnoxiously opposed to the compaction role, is that it actually plays a role in transcription itself by helping initiate transcription. So quick little video here. This is Tfam, just like you guys just saw, and it actually recruits the RNA polymerase within mitochondria, which will melt the DNA with the help of one other transcription factor, TFB2M. Okay, so that's pretty weird that Tfam's needed to bind these promoters and actually recruit the polymerase, which is kind of the opposite function if you think about the fact that Tfam also binds and compacts mitochondrial DNA. How does it choose which one to be, and how can you modify this to allow these other processes to occur is pretty crazy to me. So yeah, I'm trying to tease this apart. So yeah, again, how would a post-translational modification to Tfam affect this role of Tfam, especially in initiation, is another open question. So yeah, I've been talking about post-translational modifications. So really quickly review the one that I care about the most, and there's an endless amount of them. One of them is phosphorylate, and by an enzyme called a kinase, and it uses ATP. It donates a phosphate group to a serine residue on a protein. So this would be your protein, and now it has a serine residue. So if you guys imagine the picture of Tfam I showed binding, and Tfam gets phosphorylated, maybe there's charge repulsion from these serine residues on the DNA backbone now. So maybe that's a way to loosen up Tfam if you need one process done, right? There was a study. The study did find phospho-Tfam in cells, but they had to overexpress Tfam to do it. And then they tested very briefly whether Tfam in vitro, which means just in a tube, could be phosphorylated when bound to DNA, but they used a tiny little piece of DNA, and it wasn't circular. So nothing is similar to what a nucleoid really looks like. So that is a little primer for something I was trying to address in my work. The other big post-translational modification in mitochondria that we care about is acetylation, when a like lysine residue attached to a protein can act as a pretty solid nucleophile, and you actually don't even need an enzyme for this, just because acetyl-CoA is so prevalent in mitochondria and in the mitochondrial matrix that you can get an acetyl group on the end of a lysine. And if you had a lysine on Tfam, going back to that picture of Tfam, you would imagine that maybe this lysine helps have a favorable reaction or favorable interaction with the DNA backbone, and now you've actually lost the charge on that. So it might be a way to loosen it. And again, this research showed that, yeah, acetyl Tfam was detected in cells. They overexpressed the Tfam in these cells, so that's not quite as legit as you'd like, but okay. And then their in vitro work, actually, instead of just adding the group itself, they swapped amino acids. So if there was a lysine that was previously acetylated, they would just mutate that to glutamine to mimic this. And that's actually not a legit swap, since glutamine will have a pretty different shape than lysine or an acetylated lysine. And they did the same with phosphoserine, mutating it to aspartate. So, yep. And then this group went a step further. They actually added the acetylysine, but then they didn't quantify in a stock how much of their stock was actually acetylated. So all their results are pretty weird, considering that you don't have any quantification of how much Tfam is acetylated. So, yeah.
I had a quick question on the
perfect.
back on the slide before this.
Yeah.
If it's non-enzymatic, how does the Tfam decide how many lysine residues it wants to translate or have this acetyl group on? Like, as a chemist, I think in terms of percentages, I would just see it as like 100% or 90% or whatever, if it's non-enzymatic. Or maybe there's groups that are sterically hindered, maybe, and those groups are not added, I guess. Is that what we're thinking?
Yeah, that's a great point. Yes. So there's all these different reactivities of a lysine group on a protein based on exactly what you said, like whether it's exposed to the solvent. Some things go as far to say if an acidic residue is close but not too close, it helps with basically maybe extracting, I guess it would add, make this, a better nucleophile, right? In a sense. And yeah, I guess it would pull off proton. So yeah, if you have an acidic group a little bit close by, not to form a salt bridge with this lysine, it's more reactive. So there was 7 angstroms, but if you go any closer, then it forms a salt bridge, and then the lysine's not reactive. So it's actually super sensitive to which lysine will be reactive or not. But if there's an acetyl transferase, which there could be in mitochondria, it's not exactly known yet, be a super cool thing to tackle. But God, only have so much time. And so anyway, yeah, it's not what you would think. It's not 100%. It's actually very low a lot of the time. It can be 5%. So yeah.
Thank you.
Yeah. Cool. So what I tried to tackle, how much time? What am I at here? I'm almost done. Yeah, so what I tried to tackle is is Tfam bound to DNA, kind of much more common, compaction of a circular DNA, hopefully a big piece of DNA. Is it accessible to these modifications? No one's really even shown that, and hopefully I can show that. And do modifications to Tfam change its ability to compact DNA? And, we also want to, characterize the modifications much more, actually quantify them, right? Versus what's been done before. And then do modifications to Tfam change any other functions, like aiding in transcription initiation? So forming that initiation complex that I showed you guys. Or do they change its ability to serve as a roadblock to transcription? So if it's just sitting on DNA, it's right now known to be a roadblock. And if you modify it, and then if it can bind, even if it's modified, does it change anything? So yeah, I can stop here and continue on later, or else we'll get like one slide into my data. So it's up to everybody, I guess.
What do y'all think? Should we do a second session to finish up with Sean? We could easily do that.
Sure.
Okay.
Sean, you good with that?
I'm good with it. Yep. I think
I
that's
know it's difficult. I apologize for not getting this information from you prior to this so that we could have scheduled a two-parter. Anybody got more questions for Sean? Not right now.
cool.
You probably know how to find him. Emma, are you ready?
Yep, I'm ready.
Okay, let's go for it.
Give me 1 sec to set up. Okay, and let me know if you guys can't see this, but hopefully it's fine. Can you see the title?
Sure. Yeah.
Yep, okay.
Everybody good?
All right, cool. Hi, everybody. I'm Emma. I'm a third year in the neuroscience program, and I'm going to be talking to you today about characterizing white matter hemodynamics using a novel arterial spin labeling MRI technique. So I think, unlike probably most of you, I am in a different area. I'm in neuroimaging, and so this will be probably something different than you're used to listening about, unless you're taking one of the MRI interfaces courses. So here is a little intro. Get my mouse. Okay. All right. So this talk is probably mostly going to be about background, and I'm going to show you just a little bit of data towards the end. But I think, given that most people here are probably not in MRI, I think it's probably important to give a little bit more context. Okay, so I said I was interested in cerebral hemodynamics, specifically hemodynamics of white matter. And I'm interested in doing this in aging because there is a lot of evidence over the last maybe decade and a half that Alzheimer's disease has a very strong vascular component, especially of the cerebral microvasculature. So it's shown that dysfunction in the microvasculature, meaning small arterioles and capillaries, may play an important mechanistic role in the development of Alzheimer's disease and the progression of Alzheimer's symptoms. Hemodynamic change, which basically just means the change in blood flow in the brain and the factors that cause that change, could serve as an imaging biomarker for Alzheimer's disease, which is sorely needed because so many Alzheimer's disease treatments have failed. Hemodynamics is a potentially modifiable disease process, or not disease process, it's a potentially modifiable factor. And so it's a good target for future interventions. And further, hemodynamics is easily measured with in vivo clinical imaging tools. So it's not something invasive. It's something that you can do in the clinic, you can do in research. And so it's great to be able to follow that for several years at a time. The current research shows that gray matter hemodynamics, so in the cortex of the brain, are known to be abnormal in aging. But not a whole lot is known about white matter at this point. But though we know that white matter is especially vulnerable to the effects of dysfunction, we think that this may be an even more specific biomarker than what we've seen so far with gray matter changes. And so I want to talk a little bit more about why the white matter is vulnerable to this vascular dysfunction. Specifically, this is due to two key features of our biology. Number one, that white matter is at the distal end of the cerebral vasculature. So if you look at the image on the right here, basically the way the blood flows is it comes up from these from the arteries down here, and it flows all along the brain and eventually kind of reaches in a circular motion and then goes down into the white matter. So these little vessels along the pia here, pial vessels, those perfuse the gray matter first, which is the darker band of tissue. The white matter is only reached by penetrating arterioles. And so it just takes a lot longer for the blood to flow throughout the vascular system and for it to reach the white matter. By a lot longer, it's not anything insanely long. It just takes like an extra second, an extra second and a half or something. But when you age and when you have vascular disease, that can actually have some pretty severe consequences. And the second part of this is that white matter just has a lower vascular density than gray matter does. So it's not shown on this diagram, but the gray matter is contains many, many small little capillaries that provide blood flow and oxygen to the cortex, which is very important because cortex, as we know, is doing like a lot of the main brain processing, the brain power. And the white matter has just fewer, more sparse supplies. Okay, so as I said, the amount of time that it takes for circulating blood to reach and perfuse white matter tissue, or perfuse tissue in general, is called the arterial transit time. This transit time, just based on the biology, is longer in white matter. And as I mentioned, this is also, And as I mentioned, this is also longer in aging and disease states, and this is due to a couple different functional alterations and morphological alterations, specifically like tortuous vessels, stiffness in your arteries, blocked arteries, decreased cardiac output. So there's a lot that happens in your body that goes into this lengthening. Okay, so the tool that I mentioned that I use is arterial spin labeling. This is an MRI tool. It's a class of MRI techniques, actually, that is able to capture several features of hemodynamics. The advantage of this technique is that it is non-contrast. So what we do is we magnetically label arterial blood water, and we watch that blood water flow throughout the brain. Most ASL techniques are able to measure cerebral blood flow. There are also other techniques that can be measured, like vascular reactivity and stiffness, and there's a couple other measures. It allows you to do some pretty cool things. So this is one example. This is not the kind of data that I collect, but this is a cerebral blood flow image. I do collect those, and you can collect them pre and post. This is a hypercapnia challenge where somebody's breathing in air that has more CO2 than it should. Okay, and so for ASL, with all standard techniques, the arterial transit time is a huge challenge, even in just normal, younger, healthy populations, not even getting to aging or disease. But when you get to aging and disease, it's a lot more of a problem. So the reason that ATT is a challenge, I will explain by first just going through how a normal ASL scan is acquired. So you have the image of the brain here. This is the person in the scanner. What happens, and this is just a schematic, what happens is we tag on a large band at the base of the neck where the vertebrals and the carotids are. We tag blood that's flowing through that plane. So as blood comes up these arteries and passes through here, it gets a little magnetic tag. And it continues to flow all the way up the neck and into the brain, and eventually settles in the capillary bed where it's destined to go. And we capture, I don't know if you can see my mouse on this. It looks like not. Okay, well, sorry. Flows up through the brain. The blue boxes are where we capture the flowing blood. And so that will be our resulting image, is those blue slices. When you design an ASL sequence, what you have to do is take into account the amount of time that it takes for blood to flow from the labeling plane to the capillary bed. This is what we call the post-labeling delay. Typically in younger healthy adults, it's within a very small range, although it does vary in individuals. And if you can account for this amount of delay, then what you get is a very nice looking image on the right there, where you have accurate cerebral blood flow maps. In aging or disease, what you might see is there's a narrowing of the arteries where atherosclerosis occurs. You might have tortuous vessels, or this just might be slower for other reasons. And so if you use the same amount of delay, you're not going to be able to have all the blood reach your area that you're interested in by the time imaging occurs. And what happens is you get an image that looks like what it does on the right, where you have, where the blue arrow is showing you, you have areas of perceived hypoperfusion. So it looks like the signal is really low there, but that's actually artifactual. And you look at the arrow that's red, it looks like you have way more signal there, like you have way more blood flow. But that's artifactual too.
I guess what type of times, I don't I'm not familiar with this. Is this milliseconds time scale? Is this seconds time scale, the differences, or what? Just to kind of put it in my mind of what we should be thinking of for time scales.
Ballpark, maybe say a second.
Right.
So it's small relatively, but it is significant. So yeah, maybe a normal delay would be like a second and a half, and then a delay in aging could be, 2 seconds or 2.5 seconds or something.
Right.
So small but noticeable. Anyway, so the image on the left just looks like there's hypoperfusion and there's hyperperfusion, but that's actually just an artifact, because what's happening is that labeled blood is resting in large arteries and it's not actually making it to the capillary bed. So if you were to see the image on the right in the clinic, you might think, oh my God, this person's about to have a stroke or something, or they have something like majorly wrong, when in fact their blood flow is just slower. There's actually nothing about to happen to them. Okay. So as I mentioned, there is little that is known about white matter CBF changes in healthy aging. This is mostly because of the issues with the current techniques, mostly because of this arterial transit, which is longer in white matter and then gets longer in aging. And so there are some conflicting findings in the literature. One, that some people have found that the cerebral blood flow in white matter declines linearly with increasing age. Some people found that it decreases non-linearly, so kind of like increases in certain age groups and decreases again. And some have just found no age effect at all. And it's kind of difficult to go through the literature and get a clear pattern of change for white matter, because people use different techniques. There's a wide variety of ASL tools. And so there's not a whole lot of standardization, and then there's a whole lot of error. So my research is trying to tackle some of the heterogeneity in that literature. Most ASL studies focus on gray matter, and what we have found, which is kind of reproducible between studies, is that there tends to be a progressive decrease in whole gray matter cerebral blood flow with age. However, there's also differences in regional cerebral blood flow reduction. So for instance, the temporal and parietal lobes might have more reduction and have that faster, especially in aging and cognitive disorders, but subcortical gray matter may not have the same exact effects. Okay, so my thesis lab, my advisor and somebody else in the center, his name is Dr. Eric Wong, and my advisor is Dr. Dib Bolar. Eric Wong created this technique called velocity selective ASL. My advisor has been one that's been kind of developing the methods for it and trying to put it into clinical practice. Velocity selective ASL it is a new way of labeling traveling arterial blood. And so instead of having it like the image on the left, where it's flowing up from the neck and you have to wait for it to reach the brain, what happens is blood is actually labeled within the imaging plane itself. And the technique does that by kind of marking blood that's traveling at a certain velocity. And so you're getting blood that's traveling from the speed that it's going in large arterioles, and then seeing and then tracking the speed of blood at the capillaries. And you're kind of isolating the blood that's traveling from arterioles to capillaries. And so what it means is that the blood that you're imaging has only traveled a very small amount. So it's not needing to travel all the way up from the neck. It's just traveling from right outside the imaging plane to the capillary bed. So that's a very, very short amount of time. Gets rid of this transit delay effect. So you can see on the left here, it's kind of the same schematic. And when you would have this erroneous image at the top, now because you're labeling within the imaging slab, even if somebody has a condition like tortuous vessels, then you're not going to have the same consequence on your image. You're going to end up with a good looking image. Okay. And for this reason, for my work in aging, we hope that VSASL is going to be a better suited tool to measure hemodynamic change. Okay, so that's most of the background. If anybody has any questions, you can stop me now or just wait until the end. Just go ahead and interrupt if you want, but I'll move on to just talking about what my main projects are. Okay, so first project, these are all pretty interrelated, but just so you can see what I'm working on these days. Assess the utility of this new tool for imaging white matter cerebral blood flow. This tool has only been used at a couple laboratories around the world. It's not a very well-known tool at this point, and so there's a lot of development work that needs to be done, optimization work to make sure that our scans are getting the best possible data that they could get. Furthermore, there's not that many techniques that are able to measure white matter cerebral blood flow. So we're going to be able to compare between those limited tools that we have and see which one is performing the best. Once that is kind of validated, I will be using this technique to characterize cerebrovascular dynamics in white matter in both younger and older adults. So kind of going back to literature and seeing, okay, what is actually like, what's the normal blood flow that you should be seeing in a young adult, what's the normal that you should be seeing in an older adult without any comorbid conditions. And then I'll see how that changes over healthy aging, over mild cognitive impairment, and eventually over Alzheimer's disease. And then lastly, if I get to this, I would like to also examine the relationship between any changes that we see in hemodynamics, maybe not just white matter, maybe also gray matter, and how that contributes to age-related cognitive decline. So I'll go into a couple details about the first two projects. The second two I have not even gotten to yet. So just to give some examples of how we can kind of characterize how well VSA-ACell is measuring white matter CBF. We can do it visually by just seeing if CBF is in line with what we have seen in previous studies. So for white matter, this is typically about 20 milliliters per 100 grams per minute. That's the normal CBF unit. In gray matter, CBF is about 3 times higher. So normally we'd look for a value about 60. But there is a wide range of variability. For gray matter, it can be anywhere between, say, 20 and 70 or 40 and 70 or something. So we can kind of just ballpark guess and see, okay, are we actually getting the values that we expect to get? Number two, we can try to do different techniques to make sure that VSA-ACell is actually localized to the microvasculature and is not having any of those macrovasculature large vessel artifacts that we see in other scans. So we can do that visually by kind of looking at images like this and making sure that they look okay. We can also do this with a tool called the spatial coefficient of variation, and I'll go into that in a second. We can also look at the SNR over time because this is data that's acquired over a period of time. We can see how the SNR looks in certain regions. So we can see how the gray matter SNR looks and the white matter SNR looks. With standard techniques, white matter is typically SNR starved. Because of the transit delay, typically the label of blood actually decays before it gets there. If you broaden the amount of time before you acquire the image, that gives the label more time to actually get to the image, but the signal can decay over time. So typically, if you're even getting signal in white matter, it's usually decayed. Okay, so wanted to show some preliminary data that kind of demonstrates the utility of VSA-ACell. This is in 2 different conditions. Well, actually, the image is only in 1 condition, but historically, this has been kind of validated in 2 conditions with slowed flow. Number 1 is stroke. Number 2 is moya-moya disease, where there's kind of a pinching of, I think it's either the carotid or the vertebral arteries, but it occurs at the base of the neck. The image on the right-hand side shows adult number 1 is a patient that was ultimately found negative to have stroke. But you can see in the scan on the bottom, they do have that kind of erroneous signal of areas of hyperperfusion and hypoperfusion, as demonstrated by the arrows. Whereas when you took the same subject and scanned them with the VSA-ACell technique, it actually looks fine. So they have a slow flow condition, but they're not actually at risk for stroke. And you can compare that to the adult number 2 that was just used as a control, where they have basically the same image top and bottom. In moya-moya disease, it basically shows the same effect, where there would be kind of 1/2 of the cerebral hemispheres that is completely dark, so it looks like it's not getting that much blood flow, but that's really just because of the pinched artery. It's taking longer for blood to get there in the first place. Okay, the second way that we can kind of assess the validity of our technique is spatial coefficient of variation. And what this does is it measures the spatial distribution of the labeled blood water signal. It's very simply calculated as the standard deviation of an ROI over the mean of an ROI, and then you turn that into a percentage. This can be used as a proxy measure for the transit time, and it can also tell us whether labeled blood has reached the capillary bed or not. And so if you look at the image on the right, at the very top, you have images of cerebral blood flow. The one on the left has a low coefficient of variation. There's not much fluctuation in the signal, at least compared to the high that you see on the right. And so you can be pretty sure that all your signal is targeted to the capillary bed and is not stuck in large arteries. Where on the right-hand side, you can see that the very, very bright signals, those are the macrovascular artifacts. And if you were to just plot the CDF values that you get, you would see there's for the blue line there, you would see there's a very high number of very high frequency values. So yeah, you're basically seeing like how much does your signal vary over time, if it varies or over the space that you're looking at. And if you're looking across the whole brain and it varies a lot, you can be sure that you have artifacts in your scan. Okay, and where I'm kind of currently resting / stuck in is a lot of protocol development. So when you have an ASL technique that's not super well known, it's not used by a lot of places, or you're trying to do something new and cool, it just takes a lot of time to make sure that your data works, to validate it against different populations. In MRI and in ASL particularly, there's probably like no short of a dozen to 2 dozen different parameters that I could be changing and checking to make sure that my signal looks good. In ASL, because you're labeling blood water, it's a very small amount of the total signal that you're seeing. So you're labeling like a very, like volume-wise, a very small amount of blood and then watching it travel. What we want to do is try to maximize the amount of signal that we're getting from blood water so that we can make sure that anything that we're seeing is actually a result of our VSASL technique. And so some ways that you can do this are working on suppressing signal from other tissues that you don't care about. So for instance, you can suppress the static tissue, which is just the gray matter and the white matter, like any signal you get from those tissue types. Not anything to do with the blood itself, I just mean the actual tissue. You can also suppress signals from cerebrospinal fluid, which on ASL tend to look very bright. And when you're studying white matter, usually that signal will kind of bleed into your white matter. And so it'll be harder to see if there are any regional changes in white matter, for instance, because your signal is contaminated. And then, as I mentioned, with a bunch of parameters that you can kind of choose and test out, you can change things like sequence timing. So how long should you wait before acquiring the data? For VSASL, I mentioned that you're kind of getting blood that's traveling from arterioles to capillaries, but there's still a transit with that. It's very, very small, but there's still a transit. And so you can kind of play around in the microsecond, millisecond domain and see how long you should wait, because you might be able to maximize your signal if you wait a little bit longer, because then it'll have more of a chance to get to the capillary bed. And then lastly, for labeling schemes, so how we're actually tagging or labeling that blood that's traveling, there are different ways to do this, and there are also different ways that have different efficiencies. And you also want to make sure that you're eliminating artificial signal. So you're not tagging, or you're not erroneously tagging blood that's in large vessels if what you're trying to do is tag something that's like close to the capillary bed. So there's a lot you can do for development. And just to give you a little bit of a flavor of what some VSASL data looks like that I've collected, on the left-hand side, if you were trying, this is me trying to compare the SNR as your scan time doubles. So as you acquire more data, at what point do you get good enough data in the white matter that you don't need to acquire any more data? So it's making sure you have adequate signal in that region. This image on the left was half the amount of data as the image on the right. As you can see, the signal in the cortex, kind of the outline of the brain, that signal is pretty good in the doubled, but for white matter, it doesn't change all that much. So unfortunately, in that case, we'd have to probably keep collecting more data. And then for suppression, just wanted to show you an example of signal suppression. So you can see in the top hand side, that's just a sagittal and a coronal view of the brain from our ASL scan. When you have no suppression, you see a very like standard whole brain image. When you have suppression on the bottom, what happens is your kind of getting rid of the signal that comes from the static tissue. And so it starts at the bottom and you can see kind of dark tissue, and it gets lighter and lighter as the brain goes up. And that just helps us kind of isolate the arterial signal from the tissue. Okay. And then lastly, some next steps. Kind of give you a taste of my first 2 projects. A lot of it right now is kind of in protocol development and trying to get preliminary data before I even get to do, large scale imaging of younger and older populations. But eventually I'll work my way to doing bigger studies of healthy aging, mild cognitive impairment, and Alzheimer's. What I'm doing right now is just trying to collect data. I'm using a combination of I guess imaging at the center that I'm at, the Center for Functional MRI, and using openly available data sets. So I'm kind of starting to make progress on project number 3. We're looking at Alzheimer's and mild cognitive impairment by using open source databases and using a different type of scan than VSASL, but something that might still be able to give us some fidelity that we can see certain changes in aging and Alzheimer's. And we're switching to a new scanner system, so that'll be hopefully a better route for me to collect some data. And then after doing some prelim data, I'll kind of take a look back and see whether I want to add the following to my projects. There are a couple other options. I can look at instead of doing whole white matter cerebral blood flow change, I can see if there's any regional changes within white matter itself. I can also do that within gray matter. I could see if there's any differences in blood flow between different vascular territories in aging, which is kind of a hot topic. And then there are other additional MRI measures that I could include in to kind of get a more comprehensive picture of vascular health and aging. So I could look at the reactivity of the vessels, the compliance or the stiffness of large arteries. Those are just 2 of examples. I could also, instead of trying to eliminate the transit time from my imaging techniques, transit time, when you think about it, actually provides a kind of a valid indicator of hemodynamic status, because if you know it increases with aging, it might be just another parameter that you can scan somebody, see how much their transit is increased, and then see if that is indicative of any sort of vascular change or pre-disease state. So I can include that. And then lastly, the last thing I can do is probably stratify my patients by their risk factors for different vascular disease. For instance, if they're smokers, what their diet's like, if they exercise, what comorbid conditions they might have, like hypertension or diabetes. So there's a lot of different things that I could look at here. The first step is really just acquiring as much data as possible, making sure my tool is validated and works in a population of older adults. And yeah, I think that's about it. So thank you very much for listening. I'm happy to take any questions. Just want to thank my lab, the Bolar lab at the Center for Functional fMRI. It's a small lab. It's me and my PI, and then a former postdoc who's no longer here, but that's Dr. Woods. And then the rest of the CFMRI, Tom Luth, the director, Eric Wong, who made this technique, Rick Buxton, who's also my interfaces co-mentor, and then Aaron Jacobson and Conan Chen for 2 absolutely essential units of that team. Yeah, all right. Let me know if anybody has any questions.
That's one. Is all characterization done on human subjects, or can you do it on monkeys? Or I don't know, I guess, like, yeah, how much would transfer into, like, or even mice, or are they way too small to do any of this kind of characterization type stuff before you start getting on humans?
There's a lot of people that do ASL on mice, actually. Yeah, it's different types of scanners. There's usually like a small bore MRI animal scanner. Yeah, you can do anything that has a pulse with ASL. I think for me, I'm just interested in humans, and there's also way more logistical challenges with doing it with, like, there's not that many people that do monkey imaging, for instance, not anymore, at least, now that we have non-invasive techniques. But yeah, you can do it on anything. There are people, I think, at my center that do mouse cerebral blood flow imaging. I just have a long-standing interest in Alzheimer's and aging, so that's why humans are my people.
Any other questions for Emma?
I thank everybody for their time and for our two speakers for their hard work. And we'll be loading the video up soon. And we're working on putting the seminar series on the website. So lots more things to come. And as I said, as always, please, please, please let me know if you have some suggestions for this event or others. I'm always open to hearing suggestions. So if that's it, everybody okay? I'm going to turn off the recording and say goodbye to everyone. Thank you so much. Have a great week. Bye-bye.